How does the kidney of a bird differ from that of a mammal? There are two major differences. First, birds have a multi-lobed kidney in which each lobe has a shape like a pear or a mushroom, and the collecting ducts run around the outer margin with nephrons feeding into them. A high proportion of the nephrons are like cortical nephrons in mammals, in which there's a glomerulus and the tubule simply connects to the collecting duct. Only a small proportion of the nephrons in a bird have a long loop of Henle in which they dip down and come back. So it's only these nephrons with a loop of Henle that can change the concentration gradient in each of these individual lobes. Most of an avian kidney is at about 300 milliosmoles. Only this lower portion has regions that get to maybe 350, 400, 450, and up in some species to 600. In this region of the kidney, a bird can concentrate urine, but at best they manage a little over double the concentration. That relatively moderate ability to concentrate urine doesn't matter. The various lobes of the kidney all send their urine down to the ureter, and the ureter sends that urine into the colon, and in the colon, the urine is further concentrated. Urine in a bird enters the colon and is defecated with the feces. In a bird, the urine is high in uric acid, and the uric acid coming out of the ureters is in relatively small protein crystal complexes that fit easily through the tubes, and the proteins help them to stay round, so they tend not to get stuck. The urine is not very dilute when it reaches the colon, and birds have to reabsorb a large amount of salt and water. So I'll draw some large cells here, but of course they'd actually be much smaller. These are epithelial cells, so there would be a basement membrane here, and nearby would be a blood vessel. So let's look at what happens across these membranes. First, sodium-potassium pump moves 3 sodiums out in exchange for 2 potassiums in, and that process takes ATP. Second, because of the low concentration of sodium inside the cell, there's a co-transporter that moves sodium in because of that low concentration. It's a co-transporter, and it also moves in chloride. And that chloride can eventually leave the cell through a channel. The potassium that's being moved in by the sodium-potassium pump moves back out of the cell through leak channels. And if the bird has too much potassium, some of that potassium may escape into the lumen. Because we're having a net movement of sodium and chloride in and then out via the sodium-potassium pump and out via chloride channel, if there are aquaporins present, water will follow passively, move from this area of now lower solute concentration because we've lost sodium and chloride, and water will move into the cell, and again, because sodium and chloride are moving out, water will move out into extracellular spaces in the blood. So we have recovery of water because of 1, sodium-potassium pump, 2, that brings sodium and chloride into the cell, the chloride leaving through chloride channels, that 4 brings water in following the higher concentrations produced in the cell by this passive transporter, and 5 moving through aquaporins out of the cell. That loss of water has consequences. If this purple is uric acid, as water leaves along these cells through aquaporins, as the water leaves, the uric acid begins to form bigger and bigger crystals. As these crystals grow, the uric acid is no longer free in solution. The uric acid is now in these tightly packed crystals. The uric acid is continually removed from the solution, which means that each new uric acid that joins a crystal is no longer dissolved, is no longer a solute, and no longer will hold water behind. The final result is excretion of feces with the remains of the urine that is very dry, has these large crystals of uric acid that are the nitrogenous waste of a bird, and a large amount of recovery of water and salts for the bird. In fish and amphibians, the typical kidney is long and slender, and if we zoomed in on some part of it, what we'd see is collecting ducts with individual nephrons that twisted around back and forth over each other to join the collecting duct, with urine going down the collecting duct. So in each there would be arterial coming in, glomerular capillaries, blood going out, and twisting around. So we'd have blood in, out, capillaries along the rest of the nephron would be reabsorbing valuable molecules and secreting waste molecules,